SPIE, the International Society for Optics and Photonics, was founded in 1955 to advance light-based technology and is one of the most expressive optics society worldwide. One of its chapters is based at São Carlos Institute of Physics and run by our students. The lectures happened today are part of the chapter's activities. So, let me introduce you, Professor Dr. Igor Meglinski. Dr. Meglinski received both his bachelor's and master's degrees in laser physics from Saratov State University, Russia, and his PhD in biophysics, biomedical optics, at the interface between the university and the University of Pennsylvania, USA. His biophotonics group is a member of the Paulo Biomeding Consortium, which in turn is an associated partner of the Europe Biomeding Infrastructure Network. His research group employs several optical experimental techniques for biomedical purposes, and he kindly accepted the invitation from our chapter to visit our institute and speak to us. Professor Igor, we greatly appreciate your availability to be with us.
Good morning, everyone. And it's a great pleasure to be here. So I never was in this side of the Brazil. I was maybe on a conference. I think that also was organized partially by your team in Rio de Janeiro. And basically, that's maybe all my knowledge of Brazil. And of course, it's for me, it's a great chance to see, how the sciences works on this side. On the other hand, your research group is well known around the world, and Dr. Karachi, so she invited me several times to come, but all the time it was overlapped with something else. And now I'm okay. So as I promised her, so I'm came to your side. And today I will present the one of the project that's focused on optical diagnostics and imaging of blood and lymph vessels and microcirculation therein. But I also want to show the other project that we do in our research side. And if you see any possible opportunities for you to, to collaborate with us, so we're very pleased to, to establish a possible collaboration and so on. Okay, but I'm first start a bit more introduce myself. As it was mentioned already, what I did. But my name is Igor. And I don't know how you're familiar with the names, but if you look at that cartoon, so you will find out that actually that Igor is very scientific name. You see, so basically Igor, they was assistants of Dr. Frankenstein, and they used electromagnetic power to create new life. And I'm also using the electromagnetic power, like optical part of that, for diagnostic purpose. So the also difference that I'm not the assistant of Dr. Frankenstein. Okay. And again, I was graduated in the Saratov State University in Russia a long time ago, and then I've got like a PhD scholarship and studied and did my PhD in the University of Pennsylvania in the United States. And after that, I did some postdoctoral research, as usually we do, and then I got the docent or lecturer position in the Cranfield University in the UK, and spent there about 12 years working in this university. And then I was invited by University of Otago from New Zealand to establish biophotonics research activities on that side, and spent about 6.5 years working in there. And recently I moved to the University of Oulu in Finland. So where is that? So for example, the Cranfield University, if you know, located in about half an hour by train from London. between Cambridge, which is here, and Oxford, which is there. So it's like scientific part of the UK. So and in New Zealand, so I was lucky to work in the most southern university in the world, in the University of Otago, which is placed on the South Island and the most southern city called Dunedin. And that also is important that here is so-called Otago Medical School. And for us, it was very interesting to work there because we have direct application of optical and photonics technologies in medicine and biology. And this school is well known in the Pacific region because many physicians and medical doctors who are practitioners in there, they studied in this medical school. Okay, and now I'm working in the most northern university in the world, in the University of Paulo. And as I tell everyone, so nothing changed, just the spin is changed the direction. Okay, so it's a university located about 600 kilometers north from Helsinki, and the city is not big. It's about 200,000 the population. So, but what this city is known really that here was the biggest research and development departments for Nokia. You remember the Nokia phones. So Nokia is not working anymore in there, but the many companies that worked with Nokia as outsourcers are still in there. And we have worked a lot with these small companies that are very active in the optics and photonics research. So also, what should I say about that? So we have that faculties, and I represent Faculty of Information Technology and Electrical Engineering. And also, this university is quite young. So it's like only 50 years old, and that it's very modern. And we have about 16,000 students and about 500 exchange students per year. And thus, those of you who have an interest to be one of the exchange students, so you're very welcome to check what the programs exist. So there are basically a lot of programs for European students, but I'm sure there should be some for outside the Europe, and we have a number of students from many countries. And actually the administration of the university and government of Finland is very support and very happy when we have like exchange between the students, between the researchers, and one of the purposes that I came, if we can establish sort of this collaboration or exchange visit, so that will be great. Okay, so, and you see, so this university, it's quite colored buildings, quite modern. So what I was surprised, there are no security at all. Everything on the sensors because of working with Nokia, and it's placed in a, let's say, around some forest. So city center is somewhere there. So one of the reasons that students have no way to escape somewhere, they will be focused on the work. But of course, it's not like that. So we have a quite freedom. So that's another view. And here we have like the new development area. So this university is growing up, and they expected that we'll build the big technopark. Okay, what I was particularly surprised, so that in Finland, everyone using the bicycles. So, and this city, particularly known in Europe, that the city with the longest bicycles roads around the city, that not overlapping with the main roads. It's very safe. And you see, so you always going through the very nice parks, like on a bicycle. Even in the winter, people do that. So, and I'm also now using the bicycle, which is very convenient for some time. Okay, snow. Yes, we have the snow in Finland, and even now it's already snow, but it's not so bad. For example, when I arrived, so the first winter it was like 1.5 meters snow. And last winter it was 40 centimeters of snow. And Finnish people told me that, okay, it's something unusual, global warming, or something happened, so we have not enough snow now. Yesterday I spoke with some Finland. There was just -2 degrees, which is not so bad, and some snow. But snow is the experience. So the university have some tradition. For example, when it's February or January, so they do the special event when they make the hole in a lake, and students who want to get some investment for their projects should explain to potential investors the idea for that time, how long they can do that. So, and I think it's a good way to encourage students to provide new ideas. So now I think we might need accept in our group as well. So for example, if you don't finish your PhD on time, go to the lake and the ideas will come quickly. Okay, so this again shows what is the degrees. So we have like minus something, sometime -40, but it's again not so bad. It might be 1 or 2 weeks only. And in summer it could be +30, like here, and everyone can swim in the lakes, so it's not so bad. Okay, so now about our group. Actually, this group was established by Professor Risto Myllylä in let's say, 20 years ago, and he's retired now, and he's our emeritus professor. And I'm now take this responsibility. So that's the half of our group. We have about 40 researchers, and he is the half of them. The others teaching and do some research and for some reason. Okay, so we have like about 4 professors, some docent positions, postdoctoral researchers, students, and the colors means that. Colors means that they're some exchange students or visiting students, and we have many of them, and possibly not all of them are mentioned here. Okay, so, and traditionally, again, our team is working on the development of new sensors and linking them with wireless solutions with the modern phones, for example. That's the reason is because, of course, Nokia was there, and many businesses and many scientific research project was associated with Nokia. And we still continue that way. So you see, so we basically, in a group, we working on all kinds of sensors for medical applications, for sports, for now we also consider food, ecology, and many other applications. But again, so the one of the important parts is we want to implant the wireless devices with that solutions. And in our team, we have all the circles. So means we able to start with understanding of the process, with modeling, simulation. Then if we feel that it works, we can test it on some animals, on patients. And then we're able to produce some parts of the sensor with this small and printed electronics, light sources, and then we're able to integrate them to the particular device and use it for possible applications. Okay, also our group was recently recognized by this Science and Technology magazine, which is associated with the European Commission as the most northern team in biophotonics. And we was very proud that they published several pages describing of our research activities in this magazine that's, again, presenting the European Commission. And here the picture that we specially produced for them. Okay, we established recently the SPIE chapter and also chapter, and similar as you have here. And this was found that it was the most northern chapter in the world. So, and I can pass the respect from this chapter to your chapter. So, and if we have about total 26 students. So, and again, so if there opportunity to develop any inter-chapter events, so we can think and discuss it as well. Okay, so, and here the summary of our biophotonics research activities. So, as you can see, we have a lot of studies with the brain. And what I was surprised that when I was in the University of Pennsylvania, we developed some imaging technologies for brain activities. And in Oulu, that was also developed. And we use these devices for direct clinical applications with MRI and other techniques for just real clinical diagnostics. And we're still working on the improvement of that and for different, again, fundamental studies. So, okay, so we work a lot also with the diagnostic of tumors. So we're trying to do now, to build the new sort of tomograph for breast cancer detection. So we work a lot with plants, fresh agricultural products to find the new way how to make this diagnosis for the quality of products. And with meat, I should say as well. Skin, blood flow, biosensors, so that's one of particular interest, and I partially will discuss it today. So, of course, many of the studies associated with small animals, and you will see some results that we used for small animals, but again, they're very linked, and we use small animals like models to prove the concepts of the technologies. So, some biological tissues, like connective tissues, like joints and cartilage, they have a unique optical properties, like birefringence, for example. And of course, we also looking for new techniques that could be used for diagnosis of these connective tissues, but also for us is interesting physics behind that interaction of light with these tissues. Okay, so the infants. So, see, for example, there's someone who have a special needs. You know, when infants are born, so the blood vessels, the biggest blood vessels, or veins, for example, they are about 40 microns in diameter. And physically, we have no needles to take, for example, blood analysis from the skin, and it's painful. And for typical practice, they use the biggest vein on the leg for the newborns, and it's very inconvenient. And thus, we're looking for how we're able to do it with optics to measure some signals from the bodies. And of course, we're involved in many other applications with using some materials like nanoparticles and many others. So, but it is like natural as well. Okay, so here you can see some summary. So this nanoparticle that we use, the small animals, some brain tumors, and which it's okay, it's not working here, and skin. And many other pictures that was done with us. I also will just briefly explain one project that we have, quite international. So this is a picture of Lake Baikal in Siberia. This lake is quite big and have 20% of fresh water in the world. And it's a unique lake because it's quite deep. And it's like polyarctic lake, and there is a high concentration of oxygen. And thus, with this lake, we studied, so how the animals within the lake are changed with the climatic changes or pollution. And what we do, actually, we feed in these small fish, for example, with the nanoparticles, or microspheres that have inside the fluorescence dye. And measuring the changes of fluorescence signal, we're able to understand what happened inside these little bodies. And the particular interest of these biologists with whom we work, imagine, so in the Baikal Lake, they have a number of, let's say, shrimps that living on a different depths. And when the climatic changes happened, so the one shrimp that more aggressive will push down the others, and population is changed. And the biologists need to understand, okay, what happened when they started to, to do that. And but then, for a number of years, there wasn't possible to do any quantitative changes, and now we're able with this nanoparticles and the fluorescence dyes able to see inside. So, for example, there was for different temperature increasing, so they're possible to observe the changes of some activities within the body. So, and previously it was done like they took the one of the sample, and then they would distort that, and then we see centrifuge. They will produce something and measure that with standard spectrometry. So, which is inconvenient, and we demonstrated that if we took just this sample, we're able to see some fluorescence changes, and these fluorescence changes could show us what happened within the body. But it's not enough, and thus we suggested another idea. So we used a nanoparticle that we called like optical multisensor, and similar to the electronic nose idea or electronic tongue. So we're able to, by changes of fluorescence peaks, we're able to see what happened inside. So changes of oxygen, temperature, pH, bacteria presence, something like that. Okay, so, and that idea we started to do in the United Kingdom, and we used this approach for bacteria growth and other tests for some samples, including some urine samples in the clinics. But now we just simply introduce it to these shrimps, and it's possible to demonstrate that these particles can be localized within brain and within the particular parts of the body, and they are stable there for the weeks. Okay, and recently we just published it with in more details, like how it looks like. And you see, so this is just a live animal, and when we feed the animals with this the microcapsules, and then it's possible to see inside the body and check what the body is telling us. So our other idea is that we're talking about the sensors, but why do we need the sensors when the sensors are naturally developed? Okay, that's animal. The only thing, if the water is not good or what the quality of leaf is not good, so we need to know how to read this information from the body, and this microcapsulation might be the one of the way how to interact with these animals. Okay, so the other project that I will also briefly discuss, it's called optical biopsy. As I found from yesterday's presentation, so you have a very involved in the studies with clinical, but I want to show the other view on that side. Okay, so what is the optical biopsy? Okay, I don't know how it works in Brazil, but I will give you an idea about my experience, how it works in the countries like United Kingdom, like Australia, New Zealand, and others. Basically, okay, so when the patient is seeing that something happened on the skin, let's say, is coming to the doctor and saying, look, I have something. I'm not sure what is that. And general practitioner also not sure what is that. And what they do, they simply will cut the piece of tissue one of the way. But because they don't know what is that, but they don't want to get some risk, so they simply will cut it and put it in a plastic cassette, the pieces of tissue here. And then they will prepare so-called paraffin-embedded tissue block. And then when they will be a number of blocks from different patients. When they get this collection, they will send it to the special laboratory, and when they will use this so-called microtome, and they will slice these tissues with a very thin layers for 3 to 5 microns, and we'll put it on the microscopy slides, and that slides will send to pathologist, and pathologist will decide, is it cancer, non-cancer, or what is that? And you see, so to produce the good slide for microscopy, it's not a simple way to do. So you need to be experienced. And thus, preparation of histologic slides and this analysis, so it takes time and requires some, particular doctors to be. So, and sometimes it's not easy to make the judgment. So what is that, cancer, non-cancer? And it's required to involvement of many people. And also it requires to training of staff, young staff, to, to understand how it really works. So, but we have a number of patients. And the results, we have queue, cost, delay in treatment. And you know that for cancer treatment, it's as soon as early the treatment is started, that it's easy to maybe treat it. Okay, so that's the procedure in the hospitals. And when we started to work with these guys, so I'm asked one of the professors, okay, what you see really, what is your vision, what is there? How do you know is it cancer or non-cancer? For example, that's what they see in the microscopy slides. And I'm saying, okay, so what is that? Is it cancer or that cancer or that cancer? How do you know? And they basically told me, well, so we have like 20 years experience, like, to see these microscopies, and we know. I said, okay. I said, I have no experience at all. I said, come on, give me a secret, How do you know? I said, well, we measure, we see density, we see some boundaries and some many. But what I should say, it's very subjective. So one doctor will say that cancer, another will say it is pre-cancer, other say it might be not cancer at all. So how do we know? Nobody knows. And okay, and I thought, okay, so are we able to use light? If these people able to say that cancer, non-cancer, we can suggest a tool that could be automatically give us an idea, so cancer or non-cancer. And first idea was, okay, so if I send the circular polarized light that will propagate through this, so will the rotation of polarization changed for cancer and non-cancer differently? And that's what we started with. And we demonstrated that for thin layers, it's not working at all. But if we illuminate this sample, I mean thick tissue, we're able to see the difference in the changes of polarization. For example, here on a Poincare sphere, and that shows like non-cancer and cancer direction. And that's one of the projects that we currently working on. So we're trying to develop the technique that will screen these thick samples and automatically give us or to doctors solutions for that. And we demonstrated that it's very well works. So we're able to recognize even aggressiveness of the cancer. And for example, we use here the Poincare sphere as a graphical tool for presence of cancer. Okay, so, and the next step, we think that we're able to produce maybe some endoscopy that will be used for in vivo, so, but we're not coming to that yet. So this is what we work at the moment. So these results were published in the Journal of Biophotonics, and those of you who have an interest, so you can find it in the, I think last year, in the April issue, so the detailed description of how it really works. Okay, and now I come into this optical diagnostics and imaging of blood and lymph vessels and microcirculation as announced originally. Okay, so, but first, before we started to discuss it, I just want to start a bit to refresh your mind about some physics. So what is the coherence properties of light. How many of you remember what is that? Okay, I'll start with a basic definition for the students of physics for the second year. Okay, so if we think about the laser, what is the laser? So here is the gas laser. So the gas laser, because it's classic representation of how the laser works. And what we actually have, we have the active medium, and we have here two mirrors. And we pump with energy this medium. What does it mean, pump? We apply here the energy. What happened then? The atoms absorb the energy and produce the light. Okay, so what the energy, it could be electricity, it could be temperature, anything. That will produce the light. Okay, so what happened then? These photons will go in different directions everywhere. But those photons that will touch in the mirrors, so they will reflect. And we're still pumping and pumping and pumping and pumping. So and the more photons are coming, and some photons becomes reflecting and producing other ones. And thus, after we do it, it's many, many times. So this between these two mirrors becomes, the like motion of the light. And other photons are not here because we don't see them. And then we come into the resonance point when between these two mirrors will be perfect wave arised. And then the trick is we open here the hole and laser becomes lasing. And thus, you see the wave, which is a good wave, between these 2 points, between two mirrors, and that we called coherent length of laser light. Okay? And if we think now about the coherent properties of light, so we have this, we call it temporal coherence of light, and sometimes if we consider the laser beam, so that could be not as good as we idealistically want to be. And that's called special coherence light. And then if we check the available light sources from lasers to normal light, laser diodes, cheap lasers, so the coherent properties will be different. And for a number of years, we thought, okay, so how we can use it, which laser or light source we can use for what purpose. And why coherent properties are important? There's another 2 slides from basic physics. So imagine if we consider 2 waves, and these 2 waves interact somewhere, so we should consider the interference. So what means interference? We just should define the product of 2 waves in a particular point. And mathematically, it's easy to see that, okay, so when we do these mathematics to find out what is the product of these 2 waves, we will come to the form when we have original intensities of 2 beams and we have interference term there. And then, after some algebra, so we're able to demonstrate that, okay, so that this product will give again this, the 2 intensities, and plus this interference term that relates on the phase difference between these 2 waves. And how it works really, so imagine if we have two waves, and if they in phase, we have the addition of the amplitudes. If they're not in phase, we have, here we have constructive interference, destructive, but also we have the term all the time when they're not in phase. And that produces something new. And thus, if we think, okay, so what we detect, so we will always have the position of the detector when we have the, nothing changes, but there will be the fluctuation of the signal. And typically, in most of handbooks, we consider that part of equation. And we're never talking about the coherent properties of light, because in most of books assume that coherent properties are perfect. But here is the term that shows, like, if this coherent length is big, so this term, comes to the unit, and we always have this the part of this equation. However, if the coherence length is small, you see, so this exponential term becomes close to 0, and this one escaped. So in that case, for the small coherent length, so we have much different situation. We're not seeing something. What we should see, so when we have the perfect coherence length, the many waves together will produce some speckles. And this is a fundamental property of light, and we involved, to understanding of how the speckles can be used for diagnostic purpose. Okay, that's one fundamental issue. And the another one about the coherence that I want to also mention is the also fundamental property of coherent light is the so-called enhancement of coherent backscattering, which is typically represented by the peak. So I will just maybe show the movie. Oops. Okay, so I'm not sure, so when I checked it was worked, so. Okay, so if movie not working, so I will explain here. So what has happened, so if we send the plane wave to the medium, so the partial, some photons will propagate through one random walk because of multiple scattering. And theoretically, it can be shown that we always will have another way that will propagate on the same way, but from the different point. And thus, if we have these two different path lengths, so we will have the constructive interference, and that produces this enhancement of backscattering. And okay, so and then we can use this backscattering for this system. So if we know where to see this enhancement, so we're able to apply it for biological tissues, and we can see, you see the coherent backscattering from some parts that also could be associated with cancer and non-cancer. And okay, so thus the fundamental properties of light potentially can be used for this diagnostic purpose, and that's and this what we also work on. And now I'm coming to this dynamic light scattering that associated mostly with the speckles, as I already mentioned. So what is that? Basically, this is known for a number of years, and how it works really, the concept of this technique is based on the photon correlation spectroscopy. So we have a laser, and we illuminate our sample with anything. It could be nanoparticles, it could be scattering microspheres, blood, red blood cells, anything. And then we use the photodetector that works like a photomultiplying tube in a single photon counting, and we measure the light that's scattered. And based on these measurements, we're using this correlation approach to find out, okay, so what is inside this medium? What is the basic idea behind that? Imagine, so we illuminate the scattering medium, and light is scattered on the different ways. So if we consider the detector here, so we will see some speckles, as we just discussed earlier, and these speckles will be changed if we consider that on time and on different angles. So, and what, if we think what is really happened? So imagine we have one sample with big particles and one sample with smaller particles. When light scattered, we will see some fluctuations in both times. And basically, this technique works on measuring the intensity fluctuations during the time. And what we can think about that? Okay, imagine so I have the one sample where the particles have a Brownian motion. I'm illuminated it with a laser light, and on the detector I will see the fluctuations. And then I have another sample when all the particles, let's say, aggregated, like red blood cells do. And when we send the light through, we will see also intensity fluctuations. But these fluctuations will be different. But it's difficult quantitatively to say what is the difference. And we use correlation analysis. So it means we use these fluctuations and produce correlation function. And then we use another one and also produce correlation function. And you see the slope of these correlation functions will be different. And that difference, that slope, used as quantitative measure of difference between the particles. It could be, again, it could be motion, size, and many other issues. What is important? Imagine when we're talking about the measurements, we always measure intensity. When we're trying to represent our results, we're talking about electric field, which is not the same thing. And you see, so that's how electric field is relates to the intensity. And how we're able to then to make interpretation of the results that we measure in terms of electric field. And this is possible based on the Gaussian approximation, so known that is implemented in a so-called Seidger's relation that make a links of intensity correlation function. We see field correlation function. And this is actually the big history behind that. So imagine, so the Brownian motion of particles is known for a number of years and was first observed by Robert Brown. But he didn't understand what is that. He just saw that in microscopy. And then, you see, so 50 years later, there was like, found the relation with thermal molecular motion. But again, there was no theory behind that time. And only Albert Einstein, who suggested this mathematical theory for Brownian motion. And that actually was partially his part of the Nobel Prize. Okay? So, and actually, there is a good relation to, again, to statistical mechanics, and you can find some details in the description here. I'm just very briefly to explain. So, okay, so we know this so-called Stokes-Stein's relation, which relates diffusion constant of particles, let's say, with the temperature, viscosity, and particle size. And this well-known relation. That's from the theory. When we do the measure, the light scattering, what happened? Light is coming to the particles and scattered, and we're able to find out the scattering wave vector and find the final polarization. And it happened that this diffusion constant relates to relaxation time and scattering vector. Scattering vector means the, let's say, wavelength, let's say, an angle of scattering. And thus, we're able to measure this diffusion constant in terms of correlation function. And then it's easy. You see, so we have theoretical form suggested by Einstein, and we have the results of measure. And that's when we measure this, we're able then to relate all these quantities to each other. And thus, you see, we're able to measure viscosity, or we're able to see the temperature or the size of the particles. And that gives us a number of opportunities. We can use it for this technique for different applications anywhere. And it was used a lot. So that's on just one slide to show. You see, so when we plot the different correlation functions for different, let's say, form of red blood cells, we're able to see quantitatively, without any microscopy studies, how they changed. Okay, so, and so, okay, so, and now when we consider the blood vessels, so we see that the blood vessels are filled with different red blood cells inside. I mean, the groups, and the motion is different. And, okay, so, and also, that's well known, but when I did my PhD studies with the University of Pennsylvania, there was a group of Britton Chance, who was my research supervisor, and he actually was one of the pioneers of application of optical techniques for study brain functions. And and we started to use this technique to understand how the blood flows distributed through the brain and how they activate different brain's parts. So in the future, this device could be seen like that and can be easily, to detect what is inside. But at the moment, it's much simple. So, and the ideal technique for physicians. So we have laser source. We deliver the light source to the object of interest. It could be skin, it could be brain, it could be anything. And then collected light delivered to the computer that's associated with correlator. And thus we're able to make this analysis of what is inside, how the blood is propagated through the vessels. And that's the working device. So you see, so the one fiber, the another fiber, and we can change the positioning, and there is was a test for so-called occlusion test. Means we measured how the blood changed in this place, but we make the cuff of the arm to stop the blood. And here the correlation function shows the perfect relation. So when we have the normal conditions, we have that correlation function. When we stop the blood, so it becomes, again, smaller. If I do just replot it with a logarithmical scale, I see the perfect line of the slope. That means that correlation function have an exponential decay, which is very well agreed with the theory. And then, if we use this slope as a quantitative measure, we're able to see and correlate that slope with the actual physiological parameters. Like, for example, when we have no any occlusion, so we have roughly the slope is normal. Then when we stop the blood temporarily, so the slope is coming down. And then if we open it slightly, so you see, so the slope becomes bigger, and then we're able to observe this hyperemia, and then it's also relaxated to the normal stage. Okay, so that's what we see experimentally, but now we need to extract the information from that, right? So, and thus we started to build the theory behind that. Imagine if I have the light that coming into the medium and coming out, we're able to estimate how the light is localized between input point and to detector point. And then we know what we measure, and we know the difference in the correlation function. And then we need to find out, so how that relates to here. And, so from the theoretical point of view, we consider how the different photons propagate through the medium. And you see, so we're able to assign each scattering event with a particular relaxation time. Means, so is the displacement of scattering particles happened during the time and that position. And thus, we're able to, again, simulate these intensity fluctuations and make them linked with the results of experiment. And from the theory that we worked from also quite sufficient time, we're able to introduce here the Brownian motion, random flow, and direct flows. So that's possible to implement in the diffusion equation. And finally, so we're able to measure the correlation function changes and calculate it with our theoretical parts. And that gives us an idea to how we can plot the calibration curve to extract information about blood flow velocity within the tissues. Okay, so that's the theory, right? So but we want something realistic. And we started to calibrate this correlation function with the results of actual measurements on the small animals. That's our patient, and we found that a stable way to measure that on the that's a sort of ethical issues. It's a joke of students, of course. Okay, so, and here we come to another part of this dynamic light scattering that's called laser speckle imaging. What is that? So it's a simple theory behind that, but the difference is that we use the cheap laser source, that we illuminate the medium with blood vessels, and we collect the signal not in a single photon counting regime in a photomultiplying tube, but we use CCD camera. And the light that's scattered here is mixed and detected on the CCD camera. And then we need to analyze that image. Okay, so, and to produce the images that we're able to recognize where is the blood vessels and what is the blood flowing there. And it's like this principle is based on the temporal speckle contrast analysis. So when we do the measure in each pixel the intensity, and we use the standard deviation and intensity on the again on the each pixel. So just a bit more in the detail. Okay, so we illuminate the tissue, and that's what we have, like the speckles that coming out. And okay, so and then we collect a number of images, many frames. And then what I can do, I can select the particular pixel and go from one by one and through the time. So, and thus I will see here the intensity fluctuations, okay? So I can find the mean number of these fluctuations, and I also able to, okay, so to count the standard deviation. And then the contrast image is counted as this ratio. Okay, so, and thus we're able to, if we plot this contrast, we will transfer that image from raw laser speckle image to this picture that shows us very clearly the blood vessels. And then we're able to make some artificial colors here with the computer techniques and present it in a more elegant way to see with high contrast the blood vessels there. But again, the main idea for us was to calibrate these two techniques against each other. Okay, so I'm not sure if... Okay, so but if we consider the different parts of the brain, let's say here, there will be the different blood vessels involved. And thus, our correlation functions will have a different slope from there, and we need to link them to each other. Okay. So also that was required to design the combination of microscopies sometimes for laser speckle imaging, and we used it to link with the fluorescence microscopy. The reason is, when we can see the blood flow is much faster, and the lymph flow, if we're talking about lymph flow, is very slow, and we're not able simultaneously observe the difference. The other problem that lymph flow is transparent compared to the blood. And these speckles observation will be not good enough. But however, if we use one technique for the lymph studies and another one for the blood, so that might work. So, and again, so with light scattering imaging, we're able to produce blood vessels and see that in the different conditions. And this, we no use any fluorescence markers as we used for the lymph imaging. Also, just a few words about the sensitivity, how sensitive we are. And you see, if the animal is alive, so we have that correlation function. If the animal is dead, so the slope is changed. But we found that this slope is, the changes of the slope is, we can observe up to 2 hours after the animal death. What does it mean? So that means that there is still motion of blood within the biological tissues. So I'm not sure if it works or not. Okay, so I'm not sure why the movies are not working, but I can show it later after the lecture. So, but basically, this movie shows that we have any Brownian motion as people think of red blood cells. So there will be the direct motion of blood within the vessels. And what we recently found, so that when we observe the intensities distribution of blood in the brain after the animal death, so we will see some localization of blood within the vessels, within the particular parts of the brain. So why it's happened, I don't know. So, but perhaps that's some brain functions that we're trying to find what is there. And also, from these speckles, with using some mathematical approaches, we're able to produce a very nice colored pictures when we can able to see the intensity of blood flow within the brain. And you see, so they also shows, how different blood flow within the tissues. So that's another example that also what we can do. If we illuminate the particular vessels with an additional laser, we can temporarily stop the blood in the particular vessels and increase it in the surrounding vessels. So, and all these studies was actually focused on some tumor studies, tumor models, and this image that we're able to produce by using laser speckle imaging, there was no using any fluorescence dyes. And then by treated these images, we're able to observe the vessels, group of vessels with different blood flow. And thus, you see, in a tumor where the small vessels, we're able to see, let's say, coloring and see the formation of blood flow there. In the veins, will be different, so on capillary loops and so on. We also studied the reaction of blood vessels on the application of different chemicals. Let's say if we just drop the piece of droplet of water on so we see nothing. If the water is with some agent, allergen, so we see sometime with a small concentration, we see that some little reaction has happened. But again, if we have the high concentration, so we're able to see the vascular response, both of blood vessels and lymph vessels. Oops. Okay, so, and also when you work with animals, the animals have some, breathing or artificial motions and something like that. And it's very annoying sometimes, and it's very difficult to control. And what we found, actually, that some solutions that was known for years in astronomy, for example, when they produce these pictures of sun, you see, so there's some motions, but we can clearly see. And what we did, we just simply applied these algorithms to our purpose from astronomy. And thus, we were able to produce these pictures and observe the pictures even when the animal is breathing normally. So, and now we're trying to, again, to go further to implement this algorithm for clinical practice, for stroke treatment images and some other neurosurgeries. Okay, so again, so the other point that currently is a big trend to minimize the cost of the all devices here. So, and we're trying to implement laser diodes that are quite cheap. And you see, so if we able to implement several of them, what we can do really. Imagine, so again, one we're using for speckle imaging fluctuations, as we discussed earlier, and we're able to produce that image. But again, so another one, we can generate the fluorescence. Of course, we need to add there some fluorescence dye, and also will be produce the image of the diffusion of fluorescence through the body. So, and what I should say, the cost of this device will be about $1,000, which is very acceptable for many hospitals and doctors. And thus, again, so you see, so this raw image, and that's what we can do with laser speckles. And if we add the fluorescence imaging for that technique, so we're able to observe the different vessels, and we even more so we're able to see the veins and artery differently. And again, applying this the dynamic fluorescence imaging, so means we're also collecting all the response from fluorescence, and then we're able to find out the response of different group of vessels for that. And using this RGB approach, for example, here, if we're saying that arteries are red, veins are green, and veins that have a very late response will be, let's say, blue. We're able then to create the functional image of brain and seeing, how the vessels react for any changes within the brain. And this, like the picture that can be produced, and we was proud that Journal of Biophotonics selected this for the cover page last year. Again, so this technique is described in the, again, Biophotonics pages, so you can find this paper as well. But also we use this combination of fluorescence imaging and dynamic light scattering for separation of vessels, like for different purposes. And here is how it works. For example, from the raw image again through all this stuff to produce the colors imaging. And ideally, finally, we're able to create the images with tumor surroundings and vessels. And what is important, we're able to produce the images with the blood and lymph vessels, so simultaneously. And that's for those who involved in some biology studies and for fundamental studies within the understanding of tumor developments. It's a quite important issue. Again, so I'm more like a physicist. I'm not the medical doctor, so thus we provide the solutions for the clinical people, and they are happy to use it. And again, this also was published in the Journal of Biophotonics, when explanation of how we're able to observe the tumor or vascular network using this multimodal imaging approach. So you see, so we're able to very, very clearly see that. Okay, so, and I think I can stop this lecture at this point. So, and thank you for the attention. And you see, so what we're working on, that's just one of our project that we do in collaboration with some other European universities. And what I want to say that we still working on it, so there is a number of issues behind that. And those of you who feel that it's interesting and you're able to collaborate with us, we are welcome to discuss all this issue. Thank you for your attention.
Any questions?
So thank you, Professor, by the excellent talk today. I have one question about the speckle images. To do this kind of image, you take a laser beam and expand this laser beam, correct?
Say again?
To do the laser, the speckle, the laser speckle images.
Yes.
You should take the laser beam and expand the laser.
So the laser beam is illuminating the biological tissue. Light is scattered within the tissue, and when they come into the surface of the...
But the beam laser is not focused on the sample.
I see. Well, it's really depends. So when we use, for example, fiber optics, we don't do any focusing. So we just put one fiber and another fiber. So no focus. But if you want to do the measurements from the particular volume, from the particular vessel, yes, we do focusing. And we're focusing and we're trying to put this focus on the exact volume. And in that case, when the speckles are formed within the volume, you will also see the fluctuation. But it will be more like not the surface 2D speckle, it will be 3D speckle in that case.
But you do some scanning of this laser beam, or?
Okay, so when we use camera, we don't need to do scanning.
But it's possible to do
It's possible to do scanning, and when we just started to develop that technique, we did scanning, but we didn't use probe. We used the animal, basically. And the reason of that also, I didn't have time to explain all the theoretical issues. You see, so when I showed this relation, side-jet relation, when the intensity correlation function relates to the field correlation function, which is, the key issue of the technique. It only works when this system is ergodic. Ergodic system means that when we illuminate the light, the averaging of scattering events on time should be equal to averaging based on the ensemble of scatterers. What does it mean? So if we have Brownian motion, so I'm sending the light, it's produce scattering particles, and when I measure, so I measure the very, very average picture of speckles, because all the particles in motion. When we have, let's say, skin or biological tissues, we have some scattering centers that are not moving at all, and some, like red blood cells and lymphocytes, that are moving. And thus, this will be not defined as ergodic system. And but when you do the motions, it becomes ergodic. Thus, the scanning is important for some issues, but we also, when we're talking about the diagnostic purpose for, again, in imaging, we can produce, it's like momentum pictures that the physicians are very like.
And what is the typical resolution, time resolution? Spatial?
Okay, so as you saw, the resolution is very high because we're able to see the blood motions after the animal death. From the theory, the displacement of scattering particles that we're able to see will be around angstroms. But again, so we might be not able to measure that yet. So but nanometers and less than nanometers, I think, around that. So it's achievable.
Okay,
it's very sensitive technique. Yeah.
Okay, thank you.
Hi, Professor. Thanks for your presentation. So, what's the magnitude of the correlation time for samples?
Correlation time for what?
What's the magnitude of microseconds or?
Yes, microseconds.
Microseconds.
Yeah, it's okay. So you see, the blood flow. It's quite slow, mostly. So microseconds is okay. If you want to see, let's say, turbulence. So in a turbulence, the particles are very quick. So then you should come to this nanoseconds regime.
So What kind of camera do you use to get this result?
Again, so for this last result that we show, it's very cheap camera. So I don't remember the model, but I think we used it from Thor Labs cameras. We used it from Edmund Optics. So the cameras that, let's say, cost no more than $1,000. So because we did use the cameras that are, hugely expensive, even like that could cost like hundred thousands. But it's okay. So it's expensive, and nobody wants to buy that system then. Okay, from the practical point of view. Thus, we're trying to find out the way how to do it cheaper and cheaper. Of course, we need expensive cameras to calibrate, or first to understand how it works, and then we're able to produce with a combination of mathematics or some image processing, how we're able to do that. But it's a very good question, and what we expect now, when you work with camera, you should get the image. Image you should save on your computer. And then on a computer, you should do that processing. That takes time, especially if you take the many images. You need power computer. So now we have a new technologies like GPU, like graphic processing units, and we think that graphical processing units now can be used for very quick imaging processing, and also I heard that Hamamatsu announced that they are working on a new camera. So it's just, I don't know how you're familiar with the computer technology. So when we took the image, computer will take it through the central processing and saved somewhere on the memory. But it's always going through computer. If we have GPU, the idea is to avoid going through the central processing unit. So that images will go directly to GPU unit. And in that case, when you have number of images, you can do correlation of that images, whatever, and it will speed up quickly, and you don't need these high-speed cameras anymore, because everything will be used through the GPU. So it's not coming yet. So I know that Hamamatsu announced to develop these cameras and standards for that, but we're waiting when it will be coming to the market.
Thanks. Do you think it would be possible to build this experiment in, I don't know, maybe some confocal geometry to have a 3D reconstruction of those vessels?
Okay, confocal. Okay, so the speckles technique. Okay, so they based what we discussed on the multiple scattering of light.
Right.
When you use confocal, you're trying to extract only single scattering.
Yeah.
But the answer is it will work for single scattering. Means you will observe the one, maybe particle that move, do the motion, not many of them.
Thanks.
Sure.
Any more questions? So let's thank you again, Professor.